questions from online.
Yeah.
This one is a question for the whole panel. ALS is complex and needs a strategic approach to solving. What, in your view, should that strategy or approach be?
Looking at me, yeah. Yeah, so I think, so the crucial term here is complex. And complex means, it's murky, it's not very hopeful, it's maybe we cannot understand it. And I think there are good indications. I had some slides in it that ALS is not really complex, but it's more simplex. Well, it's a bit silly maybe, but so it's not simple like a Mendelian disease, but it's certainly not as complex as all these common diseases with hypertension, schizophrenia, with thousands of SNPs doing something, and then still you cannot explain it. But Amar Al-Chalabi from King's College really showed that there's good data out there to suggest that, like cancer, ALS is like this multi-step process. Maybe 5 or 6 steps are needed, which could be 3 mutations or 6 mutations or 2 mutations and some environmental triggers. So in that sense, I think there's reason for hope because it's not too complex. But in the end, yes, so it means international collaboration, pooling data, and sharing data on all sorts of levels. And that's technically, it's possible, and we're already doing it to a certain degree, but can even be more extreme probably.
So from a non-genetic point of view, I certainly would agree with the question, the premise of the question, which is that ALS is complex. The question about coordination, I think the question is perhaps asking, are people working together? And the answer is yes. And there are several national and international collaborations. In the US, there is a clinical research group called the Northeast ALS Clinical Trials Consortium, which reflects 120 ALS clinical investigators working together. In Europe, there's NCALS,
yes.
which is a similar collaboration, and NCALS and the Northeast group share many of the same measures. I do think that there should be a word of caution. Sometimes collaboration or coordination means sort of central governance, and I think in science that's not always the perfect way to go about things. I think that there has to be encouragement for individual investigators to have creative urges and think new thoughts, and the support for them to pursue those lines of investigation. I actually think that in the ALS community there's a good combination of coordination and collaboration, as well as individual opportunities for creativity. So we haven't gotten there yet, but I don't think the reason is that there's been a lack of coordination.
Okay. I'd like to make one comment about the issue of complexity as well. ALS, you can look at it in many different ways. It's a many-headed beast, but it's a very variable disease. As all of us know, you can fill up a clinic with ALS patients and none of them look the same. And so what I plead to the ALS community, not just from the researchers, but from the patient and the families, is that we need people. Participation in ALS trials, especially observational trials, and there are several going on around the world. Several in the United States, one is called Answer ALS. There are these biomarker trials, and they're not necessarily treatment trials, but they're trials to try to figure out this complexity of this disease. Why do people look so different? What are the markers that we can discover that makes us think that one person's going to have either this course of disease or another course of disease? And traditionally, relatively few patients with ALS participate. And we need to have more and more people. We can do lots and lots of animals, but the animals don't really have this disease. They are just animal models. The only real model of this disease that's true is human beings with this disease. And so we need help. We need participation from families and from patients with ALS to participate with us to try to find answers to this disease.
So one other online question. Why don't legitimate stem cell scientists and clinicians do more to expose the bogus stem cell purveyors, or what would your suggestions be?
I guess that one's directed at me, considering I'm legitimate. This is not necessarily a particularly specific problem for ALS, but it's me throwing stones at somebody else. Actually, they throw stones back. I am happy to speak in a forum like this and answer specific questions. I don't think it's useful or productive for me to take on a crusade against these folks. When I come up against them with my individual patients, I counsel my patients about this. And I think Rick Bedlack has done a very good job in putting together ALS Untangled, which I presented here today, to try to put some science behind investigations of these claims out there. So I don't think it would be productive for me to do it individually as a crusade. I think the ALS community has to do it in general.
And then this is a little bit thin ground, but I would say that that question also can be extended to the world at large. I'm
Talk about politics.
I am. I think the recent election in the U.S. is an example of facts maybe not making as much difference in emotional decisions. And so I would say that could be translated into a plea to individuals to consider facts and consider evidence, and we can all do that in our daily lives.
Are we going to speak about global warming next?
Sorry, is it called ALS in America? Is it?
Yes.
It is. It's not. Yes. So, is Lou Gehrig's disease? Is that a different, or what's that?
No, it's the same disease. So it is often true that if I'm seeing a patient for the first time and making a diagnosis, and I'm saying, well, because of these reasons, I think you have ALS. There may not be a flash of recognition with the family or the patient, but if I say, oh, it's also called Lou Gehrig's disease, they'll at least have some familiarity with it. So, we talk about it as ALS, but there are certainly some patients that are much more familiar with the term Lou Gehrig's disease. Lou Gehrig had classical ALS, and it was an early and very prominent person with the disease.
Thank you. If you talk about ALS as a spectrum of disease, do you consider PLS part of that spectrum, or is it a separate disease, and as such, are PLS patients eligible for ALS trials?
Personally, I consider it part of the spectrum of disease. And clinical trials are designed to, for the most part, because of what's called power analysis and trying to get an answer at the end, you try to get a relatively homogeneous population. And sometimes PLS patients will be excluded from that, but not because we don't, at least personally, we don't think that they don't have a motor neuron disease that's akin to ALS, but they may not fit the profile that will give us useful information at the end of a clinical trial. I don't know what you guys think.
The trial will take very, very long in PLS, so that's the practical reason. And probably if things are effective in ALS, they will be translated to PLS. But you're right, it's kind of always like we're not interested in the disease or something. That you're right. So
I appreciate the numbers are very small, but I suppose what I'm trying to get at is, do you think what you're looking at or researching into in ALS will be translatable to PLS?
I mean, personally, I treat all of my PLS patients with riluzole. There's never been a trial of riluzole in PLS.
We tried it. But, okay.
Never. I think we all agree on that.
I'm Kathy Collett from Indiana. In the United States in the last year, we saw a royal ton of work done on clinical trial guidance and guidelines and things.
We certainly did.
Do you anticipate in the next 12 months that any clinical trials will have really different designs from what we've seen in the past?
Well, clinical trial design is an evolving science. And I know from my perspective, and I was involved in those efforts, is that what those efforts I hope will be successful in doing is encouraging regulatory agencies to view newer designs and potentially more efficient designs favorably and with an open mind. Unfortunately, the nature of such consensus efforts are not necessarily to discover new information. It's mostly a collation and a political or advocacy effort as much as anything else. But I'm hopeful that that advocacy effort will help in terms of making the new designs that people are thinking of all the time more acceptable, at least in early and middle phase trials. So I hope so.
So maybe to add to that, so maybe again about genetics. Sorry, I'm biased, but maybe we need to also stratify on genotype. You know, maybe C9 patients do differently with riluzole or lithium or whatever than other patients. But
Yeah.
When a patient gets diagnosed with ALS, I thought there's a possibility that the disease cascade may have already been rather far gone for a very effective intervention. So assuming that a person presented with fasciculation or other earlier symptoms of ALS, as soon as the symptom happen, what will be your recommendation of intervention? Because I thought this would be the best time for intervention, at least to delay the gap between the onset to diagnosis.
So I think most people would agree, although we don't have great data to suggest that this has to be true, that the best time to potentially to treat somebody is as soon as you can make the diagnosis. But that, at the moment, is not so easy. And so there are many diseases besides ALS that cause fasciculations and fasciculations only. So you can't regard somebody like that as having ALS. But there's continuing very vigorous work to try to find early, potentially blood markers or other kinds of fluid markers that will give an accurate diagnosis before we might be able to make the diagnosis based on strength and coordination difficulties. I would say, though, I think in some ways ALS, we may be at somewhat of an advantage in ALS as compared to other neurodegenerative diseases. For example, it's clear that in Parkinson's disease, the part of the brain that causes disease is extremely damaged by the time the first symptoms occur. The same thing is true in Alzheimer's disease. In ALS, because the disease may begin so focally, so with an arm or with a leg or occasionally with the face being involved, but the rest of the body being really quite uninvolved, I think there potentially are opportunities if we can find a way of arresting the disease that the body itself, when you make the diagnosis, is a combination of late disease in certain parts, but very early disease in other, and maybe no disease in other parts. And so I do think we have opportunities even in the situation we're in now. But I agree with you that one of the big efforts that currently is ongoing is to find ways of accurately making the diagnosis as soon as possible.
So just to add to that, I think there's a risk-benefit ratio there that needs to be calculated. So if you have a treatment that has very low risk, meaning there are very few side effects and probably won't do any harm, probably it's easier to jump in and treat somebody early as opposed to waiting for them to have definite disease. So in my clinical practice, if I have somebody come in and I'm not quite sure yet, I start them, at least I ask them about starting riluzole anyway, just in case, because I don't think there's a real downside to that. And we know that there is a small but true upside to it. But we could go back to the stem cell thing. Would I inject stem cells into the spinal cord of somebody who I wasn't absolutely sure had ALS? Probably not. So I think you have to think about what treatment you're thinking about, and that's going to be very important for prevention genetics, because if you're carrying a gene for C9orf72 and you don't even know if you're going to get the disease, and maybe you won't get the disease for 20 years or 30 years if you do get the disease, what's the toxicity of the drug you're going to give? How expensive is it? And how long do you need to give it to prevent the disease from happening? So these are very, very important but very difficult questions.
Good. Wait.
I think the early intervention I will recommend is meditation and probably checking the vitamin D level in the blood and to see whether they're low, because that affects patients' sleep. And as a result, that can lead to fasciculation and weakness and a whole host of other problems. Thank you.
Thank you.
With sporadic, there's two types, the sporadic and the familial. If you had one of your mother or father had the sporadic, does that put the rest of the family at increased risk?
Yeah, that's a great question. So we exactly know what that increased risk is. So the lifetime risk of ALS is 1 in 300, meaning that we all have a chance in 1 in 300 to get ALS, 1 in 350 maybe. So every time you enter a jumbo jet, one person will die from ALS. So that's quite shocking, actually. But okay, so if one other person in your family has ALS, that is raised by 0.03%. So instead of 99.97% chance of not dying from ALS, you have 99.4% dying of not dying from ALS. So yes, there is a significant increase, but it's not something to worry about. You still have over 99% chance of dying from something else than ALS. That's the answer. Yeah.
Hi, my question is for Dr. Glass. So you spoke about different stem cell types in your presentation, the mesenchymal cells and the neural cells. So I have actually two questions, one about the stem cell type and about the route of delivery. So you majorly discussed about the intraspinal route, but there are other routes like intrathecal, intra-arterial. So what would you say, which would be a preferred, or can you say at this point of time, which is a better route of administration and which kind of cell would be a better cell to be looked at? From the point of view of neural cells, which are more, so they have more targeted progeny, like more specific progeny. But if we look at more primary cells, which can grow into, say, nerve cells and the blood vessels and give a more of paracrine effect as well. So what would you say about that? Which would be a better type of cell and a better route of administration?
That's an extraordinarily important question, but it's very, very difficult to answer because we don't have any data. And so right now there are multiple trials going on. There are intraspinal injections of both mesenchymal stem cells coming from bone marrow, and also of neural stem cells coming from fetal spinal cord. So those are both going in the same way. There are blood-derived cells that are being injected into the spinal cord as well. I think there are other sources of mesenchymal stem cells being injected into the spinal cord. Now, if you want to talk about into the spinal fluid, there are mesenchymal stem cells being put into the spinal fluid. That's all I know about in terms of spinal fluid, but there are not just mesenchymal stem cells. There are mesenchymal stem cells that have come out of a patient, then been manipulated in vitro in the laboratory for a month or so with some kind of special sauce that we don't really know what it is. And then they're being put back in to the patient's spinal fluid and also into their muscle. And so it's an extraordinarily complicated question. It's being hit from many different directions, and I just don't know the answer. Until we actually see either a very positive effect or a very negative effect, so we can say we're not going to do that one anymore, we're not going to know the answer to that question. It's been done in many different types of animal models as well, but again, the data, it doesn't actually say this is the best way to do it.
So just to add on to that, in your opinion, the future research should be targeted in which direction, considering there are so many options available?
You're pushing me into a corner. I think the research should continue the way it is so we can test these different ways. Now, I will continue my research based on my particular interest and my hypotheses and my biases. Other people will do their research based on theirs. To tell you the truth, though, I think we haven't talked about this today, but I think the next step in terms of, quote, therapies for ALS to delivering types of products may not be stem cells. They may be viruses. Viruses that you can package different genes, you can package different proteins, and you possibly can get them to your specific cell population in a much safer and more efficient way than actually using stem cells themselves. So I think the technology and the science is progressing to the point where we might not be talking about stem cells in 5 years.
So we more online questions. So this one may be a good Dr. Shefner question around clinical trials from a spouse and caregiver. Just really expressing frustration on how clinical trials work. Certainly the wait to potentially get into a trial, and then when you're screened, you no longer qualify. And the second piece of that is around really finding out what the results of trials are. So if the panel could speak to that, be great.
Sure. Well, I certainly understand the frustration that many patients and their families experience around clinical trials. It's interesting, whether I mean, there are many of them, and they include, you know, what was just said, the wait to get into one, how inconvenient it is to get to a study center to actually participate, criteria that are so limiting that you think that you're a typical patient, but there's something that keeps you out because of some particular criterion that you don't meet. Some of these, I think, are hard to change, and some of these things are really important to address. I think that clinical trials, it's very important to recognize, are experiments, and so they have to be designed with the highest chance of getting a clear outcome in mind. The worst thing that you can do is do a clinical trial that maybe includes lots of patients that are fast progressing and slow progressing, and because of that, come to an answer that's inconclusive and gets you no farther to a cure than you were when you started. And so those kinds of frustrations, I think, the only thing we can address is education and say why we're doing what we're doing. Other things we can do better. And so, for example, we ought to be able to design a system that if there's an ongoing trial and you meet the criteria and you're eligible, that you get in right away and that the trial starts. And one way to do that is to increase the number of clinical trial sites around at least the US and in places such as Western Europe where the healthcare systems are relatively advanced. Make sure that that we work as hard as we can to make these trials accessible. Another possibility, which I'm personally interested in, is to try to work out ways especially for drug therapies that have limited side effects, to allow these trials to exist almost independent of a study center, to have a visiting nurse come to assess problems, and a way to measure how you're doing at home, for example, would allow people to be in trials even if they were very, very far away from study centers. And so I think we should be working on that. I think the last question was, part of it was frustration about hearing about results. I think that's something we should never, I mean, that's fixable, and that it should never be the case that a patient participates in a trial and then doesn't learn what the results are. In the consortium that I've been part of leading for many years, the Northeast ALS Consortium in the US, we make sure that as soon as the trial is finished and the results are analyzed, we have letters that get sent out to every patient, but we also have a computer-based webinar where all the patients are invited, the results are discussed, questions are answered, and I'm sure we can do better, but we do try very hard to make sure that those results are disseminated as much as possible. And I think that should be the burden that anybody who does a trial accepts. There's no reason to not have that happen.
Okay, another online question. Currently, what is the main barrier to finding a cure?
We don't know what causes the disease. And when you're shooting at targets and you're blindfolded, it's hard to hit that target. I always use the example of HIV. So HIV was an incurable disease until they found the virus. But then they could shoot at that virus, and they've been very successful in making that terminal disease into a chronic disease. I think genetics, Jan can speak about this, is one of the ways to find targets. But we need to know what we're shooting at before we actually, that's why it's so hard, I think.
And the one, before we talk about genetics, I mean, it's been brought up in this room, is people have suggested that ALS may not be a single disease, but that it's a spectrum of many diseases. And that question reaches almost religious kinds of significance, because people argue passionately back and forth. But until we know what causes ALS, or what causes the many versions of ALS, we don't know the answer to that either. And so we're in the middle of this story, not the beginning.
Yeah, maybe to follow up. So of course, we need to know, we need to learn those causes. And I think the real barrier, and maybe it sounds cheap, but it's money. And it is. And it's not lack of expertise, it's not lack of patient willingness or family willingness to participate, it's not lack of smart brains that know things to do. It's about, and for example, the HIV example that Dr. Glass gave, is really because patients demanded that these therapies got developed, that that this money was really spent on this retroviral therapy, so patients were key in really get up on the barrier and really demand that this money would be dedicated to finding this chronic disease treatment. And so I think also patients are needed to mobilize funds and organizations and awareness, and so yeah, that's it, I think.
Just to speak to that point, I just came back from Italy, where we had a very large grant review session, and nearly 70 grants were reviewed. And I would say if there was all the money in the world, 35 of them were worthy of support. So I mean, they were written by thoughtful people. That if the studies had the money to go forward and get done, there would have been a high chance of useful information coming from them. But the situation was such that less than 10% of those grants were funded, and that's a money question. So I agree that that's a significant barrier.
This question might be a bit off topic, but I was just wondering, if a patient presents themselves to a clinician with fasciculations and fasciculations alone, is it possible to distinguish between fasciculations that are benign and fasciculations that could be representative of the early stages of motor neuron disease? It's my understanding that somebody would have to have some sort of muscle atrophy in combination with the fasciculations for that to develop onto motor neuron disease. So just wondering if that's the case.
Yeah, so that, I mean, it's a very interesting question. The question is, are there benign fasciculations versus malignant fasciculations? And the answer probably is no. And so this was a question that's been addressed like over and over again in physiology laboratories and in clinical physiology laboratories as well. And just in terms of the level of worry, probably the best study was done in 1987, where upwards of 250 people presented to the Mayo Clinic in Rochester, Minnesota, with muscle twitching, with fasciculations. And so what they did is that they evaluated these people very carefully. They did very careful neurologic exams and did EMG nerve conduction studies. They did the study with looking at the muscle with a small needle and doing nerve conduction studies. And then they followed these patients. And so the answer was that if you had fasciculations, but you had a normal neurologic examination and a normal EMG, except for finding the fasciculations, you had a 0% chance of that 250 group of patients of developing disease over the next 10 years. So that's, I mean, so that's not really an answer, because I don't know if there's a particular kind of fasciculation that is more malignant. Nobody does. But in general, if you have fasciculations only, but no weakness and normal physiology, your chance of developing disease is incredibly low. And by the way, most of those 250 patients were actually medical students, because they're the ones who were actually the most likely to notice fasciculations and get worried about them.
So I get fasciculations all the time. We all do. Fasciculations are part of being human and living under the force of gravity. So fasciculations themselves are not. Having said that, I have had, and I don't know, this is a little off topic, you're right, but I've had several patients who have just widespread fasciculation, I mean, everywhere, all the time. And they have no weakness, and their EMGs don't show anything other than that. And I worry about them. And some of them I've followed for several years, and a few of them have actually developed ALS. So I think it also depends on how prominent those fasciculations are. Most of the fasciculations that are benign, they come and go. But if they never go away and they're all over the place, I think you need to worry. I don't know what your thoughts are.
Yeah, well, yeah, so I think there's also this strange correlation between the degree of fasciculations and the aggressiveness of the disease. So it's not always that people with many fasciculations have a more aggressive form of the disease or the other way around. So that's maybe also an indication that it's not automatically saying that you're on to develop ALS. Yeah, we say the same. So if the EMG is normal, neurological exam is normal, just fasciculations, maybe not as widespread, then your background risk is still 1 in 300, the number I just gave earlier. So that's what we all have. Yeah.
Well, one more online question, Dr. Veldnik, this may be a good one for you. Do you believe that gene therapy could be a valid therapeutic approach for ALS?
Yes, sure. I think that goes without saying, yes. And there are many forms of gene therapy. So one of those is the antisense approach that already was mentioned. And so that typically occurs if the protein is getting toxic, so you want to lower the toxic allele. The caveat is that if you lower too much, you might induce some other pathology. For example, C9, we know that if you knock out C9, you get autoimmune disease or lymphomas, and so probably not a good idea to completely abolish C9 expression, but you can do it very targeted at the mutant allele, so that's very exciting. And there is this novel CRISPR-Cas9 technology, that you really can repair DNA, which is obviously a bit more sensitive maybe, but then again, it can also be applied in a somatic way, for example, only in the brain or only in muscle or only in the organ that's primarily affected, that you can repair the C9, for example. You can excise it and put in a wild type form of it, and that's really, really exciting. So without changing your germline, and so it's not changing the DNA of the human species. So that's really definitely.
You should maybe give the example as well, the spinal muscular atrophy.
Yes. Oh, yeah. So exactly. So the neuroscience meeting was just 1 month ago, and that kind of remembers the earlier question about the need of intervening early. So this exon skipping therapy that's going on in SMA, for example, is spectacular. People get better, and even so SMA is a childhood onset form of kind of motor neuron disease. Well, and so even if kids have this for years, people and they can get better with this therapy. It's really spectacular.
So just to maybe a little more clear, so there's two things that gene therapy can now do. If the mutation is producing something that's toxic, an abnormal protein, you can turn off that gene and lower the amount of protein. The example for SMA is the opposite. If the gene is not making enough of what you need to stay alive and healthy, you can actually develop something that will allow the gene to actually make something that works, make a protein that still works even with the mutation. So you can both replace the protein that is missing as well as get rid of the protein that's hurting you. And I mean, that's remarkable, and it's new in the last few years, and I think will be revolutionary, not just in ALS, but in many other diseases.
And it speaks to the previous question about why is ALS so hard. The thing about spinal muscular atrophy is we know what the problem is for the most part. It's a gene and a protein called survival motor neuron, and so if you can affect that gene and make more of the SMA protein, you've done it. In ALS, we're not sure what we're shooting at still, and again, Jan's going to figure that out pretty quickly, and so we'll be able to do the same thing, right?
Yeah, sure.
You just said that it needs a lot of money to solve or to find the cause. In HIV, in a relatively short time, there was a huge amount of money to get where we are now, and there was a lot of money from the governments, I presume.
Yeah.
Was that the case, and is that also something we could do to go and stand on the barriers and say, listen,
So-
you helped HIV, and we're here, we're a small group, but we have exactly the same problem.
So I have to say that I would want to be a little careful here. AIDS had a huge amount of money and advocacy thrown at it, and I don't mean to be callous, but in some ways they were lucky. It was one cause, it was one virus, and so that they found a single event that they could attack with their resources. We would all like more money, I completely agree with Jan, who said that with more money we would get to effective treatments faster. But it's also that it is also brain power, and it is also looking at a whole slew of different pathways that haven't been looked at yet. So I think we're in a little bit more of a complicated situation. On the other hand, if the ALS community can be even more organized than it already is, and I think you guys are already pretty organized, and we can advocate more strongly for more funds, that would be wonderful.
Wrap up. I just want to thank you all. It was a great afternoon, and thanks very much, and continue doing the good work.
Thank you.
So everybody, that's it for this afternoon. So thanks for coming. It was great to see you. And as you know, the symposium is running until Friday, so there's going to be a lot of positive discussions, I suppose, over the next few days. So thank you very much, everyone.